Good afternoon, and thank you for inviting me here to tell you a little bit about our story involving the establishment of what we now call the International Institute for Nanotechnology and our 20-year journey that has led to a lot of cutting-edge discoveries in science, but also innovation and commercialization in the area of nanotechnology. The IIN is an umbrella institute that covers really all research in the area of nanotechnology, spanning both campuses, the Evanston campus, which is science and engineering predominantly, and the Chicago campus, which is focused on medicine. It unites over $1 billion in terms of research, instrumentation, and educational infrastructure. And it really acts as a mechanism and an entity to attract some of the best and the brightest from all over the world to participate in nanotech endeavors. It consists of many world-class faculty from all over the world, and 2 scientists have become Nobel laureates since the inception of the Institute. The most recent being Fraser Stoddart, which I think was really an exciting point for the field and certainly validating point for Northwestern's effort in nanotech, but I think the world's effort in nanotechnology. But most importantly, it acts as a magnet to attract some of the world's best scientists to Northwestern to focus their efforts in the area of nanotechnology. And today I want to talk a little bit about some of those contributions in areas spanning a variety of very important fields. These researchers have made groundbreaking advances in many areas spanning nanomaterials and devices, nanotechnology solutions to environmental problems, ways of creating safe food and clean water, new approaches to harvesting solar energy and converting solar energy into other useful forms of energy, and then the development of new constructs that are changing the way we track and ultimately treat disease. Now, at the inception of the Institute, we knew it was important to not just focus on science, but to focus on the development of technology from scientific discoveries, and also to put in place a machine that would allow us to take promising technologies and translate them in the form of commercial startup entities. And since its inception, the IIN has spawned 24 new companies spanning many different areas of research and development. I'm going to give you a few examples of these just to talk a little bit about impact and to give you a feel of the scale of what's going on, and also some of the successes with respect to the development of real products that are now sold and used all over the world today for many different purposes. One of the early successes of the Institute was the founding of Nanosphere in 2000. This is a company that went public in 2007, ultimately licensed 175 patents in the area of nanotechnology from Northwestern University, and those enabled the development of rapid genetic point-of-care medical diagnostic tools. Nanosphere was acquired by Luminex. Luminex is built upon the platform, and that platform, which is called the Verigene system, is now sold and distributed throughout the world. The Verigene technology was based upon a very simple but powerful and elegant discovery, and that was that if you use particles in a range of oligonucleotides on their surfaces at high density, you can create spherical forms of nucleic acids that have properties that are completely different from their linear analogs. These include the ability to recognize and bind to targets much more tightly than the same oligonucleotides free in solution. It's very general in the sense that different types of particle cores can be used. That's going to be important. In the case of the Verigene system, we relied on gold cores, but as you'll see in the therapeutic areas, we actually utilize more biologically benign cores, such as liposomes. They show increased stability in complex matrices, meaning the oligonucleotides do not degrade as rapidly as linear oligonucleotides, minimal immune responses, and the other thing is these types of structures will enter cells routinely. So one way we used spherical nucleic acids was in the development of new probes in biological labeling schemes. In the case of the Verigene system, we rely on gold nanoparticle-based spherical nucleic acids. We utilize their tight binding constants to capture target DNA strands associated with disease, and we do so in a microarray format so that the particle can be used to localize oligonucleotides on a specific spot in a microarray so that we can then visualize based upon the colorimetric properties of the gold nanoparticle probes. But we also take advantage of one of the unique properties of nanogold, and that is that it's catalytic with respect to the reduction of silver-1 in the presence of hydroquinone. So if we flow ordinary photographic developing solution over a chip that's been treated in this manner, anywhere where we've captured target, we can amplify the signal by many orders of magnitude and then visualize using very low-cost readers the results of the test. Each spot in the array can be a marker for a different disease, and you can see how this leads then to the ability to create panel assays for lots of different disease states. That advance, which was first reported in a science paper in the early 2000s, has been turned into what is now referred to as the Veregene system, and as I said, sold by Luminex. Now used by over half of the world's top hospitals, and used for detecting many different disease states spanning bacterial infections, viral infections, cardiac tests, human genetic tests, and pharmacogenetic tests. It also fuels research. So this is a really important point. New technology drives new research capabilities, and we actually utilize this type of technology in our own labs to identify new markers for disease. Diseases spanning things like Huntington's disease, Alzheimer's disease, and many different types of cardiac ailments. Now, Nanosphere is a nice early example, but another powerful example of the use of spherical nucleic acids came with Execure. My friend and colleague Mark Ratner, who actually helped start the Institute, used to like to say that new materials lead to new capabilities. And the fastest way to new materials is through miniaturization. Every material, when miniaturized to the nanoscale, or when restructured on the nanoscale, has new properties. And I think the spherical nucleic acid advance is just a beautiful example of that. One of the most powerful structures ever discovered, ever synthesized by chemists, the oligonucleotides, when restructured on the nanoscale, have new properties that are enabling across many different areas of science and technology development. Execure utilized spherical nucleic acids to develop a powerful class of medicines based upon nucleic acids that work throughout the body. And they took advantage of the fact that these types of structures have different biodistribution properties and have the ability to engage targets in powerful new ways that open up the ability to create new types of medicines that can treat many diseases spanning many forms of cancer and neurodegenerative disease. Execure is a company that was founded in 2011, also based upon technology out of the Institute. It listed on the NASDAQ in 2018, became public, and now has 6 drugs in human clinical trials. Very exciting development, and shows you how rapidly nucleic acid medicines can be taken into the clinic, something not possible with small molecule and oftentimes biologic drugs. I mentioned that spherical nucleic acids have this unusual property in that they are recognized by almost all cell types other than mature red blood cells and rapidly internalized. Linear nucleic acids are not. And when you make that discovery, you want to ask why, and we did, and we figured it out. It turns out to be due to structures called scavenger receptors that recognize and bind tightly to spherical nucleic acids and internalize them via caveolin-mediated endocytosis. That does not happen well with linear nucleic acids. As a result, you now can move large payloads of nucleic acids in spherical nucleic acid form into many different cell types. That opens up the ability to think about treating disease in new ways. Now, with any technological advance that leads, for example, to a business, there's something called the Gartner hype curve that can be applied to analyzing it. And the Gartner hype curve basically breaks the development of that technology into 3 phases. The first is what's called the peak of inflated expectations. In the early days, people get really fired up. In the case of genetic medicines, which again arguably started over 30 years ago, people thought we were going to wipe out disease as we know it. You reach what's called the peak of inflated expectations. And then what happens is critical scientists begin to look at possibilities and some of the hurdles, and they turn south on it. And you run into what's called the trough of disillusionment. But if there's something there, critical scientists roll up their sleeves, work through it that are believers, and ultimately develop capabilities that are called the plateau of productivity, oftentimes lower than the peak of inflated expectations. That's been true in genetic medicines. We've gone from the development of nucleic acid medicines for wiping out disease as we know it to predominantly developing platforms that can address disease in the liver. And the reason is that's the low-hanging fruit. That's where nucleic acids go if you systemically inject them. With the discovery of technology like spherical nucleic acids and the recognition that these provide privileged access not just to cells but different tissue types, we can begin to think about utilizing this technology to raise the plateau of productivity for genetic medicines, to not just treat disease in the liver, but do so in the skin with creams, in the eyes with eye drops, in the brain, for example, when intrathecally injected. We can get to different parts of the brain and begin to address different types of neurodegenerative disease in ways that we can't with conventional nucleic acids. And even in the case of immunotherapy, where a local injection can engage the immune system and using the lymphatic system, create a system-wide response and create new and powerful forms of, for example, cancer therapies. And that's exactly what's happening. And that's really quite exciting. So when we started Execure, we recognized this and we began to think about how we could create a robust platform in oncology, inflammatory diseases, and neurological and genetic disorders, how we can span the immune system, skin, brain, eye, GI tract, lungs, and liver in ways that you can't with conventional nucleic acids. And that's led now to the development of a set of drugs, shots on goal, that are being tested for Merkel cell carcinoma, head and neck squamous cell carcinoma, melanoma, Friedreich's ataxia, spinal muscular atrophy, just to name a few. And so this is really exciting. This is basically taking nucleic acid medicines to places they've never gone before. And I think we're going to see a lot of really powerful drugs come out of this particular effort. So it's not surprising that Execure's clinical pipeline actually spans these advances. Neurology, we've got 2 drugs there. Immuno-oncology, we've got a drug called Cavro that's showing a lot of promise in treating patients with Merkel cell carcinoma, a deadly disease where there are no real good treatments for the disease. And we're actually seeing the elimination of tumors in certain patients, which is quite exciting. Dermatology, the ability to apply spherical nucleic acids to the skin in creams, allow you to treat terrible diseases like Netherton syndrome and really critical problems like hair loss disorders. Very exciting and I think very, very impactful. Okay, those are 2 really nice examples in the area of medicine, but nanotech is much broader than medicine. And I want to talk about a couple of other companies that are doing some really interesting things. Stokia is a nice example. It's a company that was recently launched, again, out of Northwestern, based upon advances there. And it's focused on materials by design, or creating tools, discovery tools that allow you to accelerate the interrogation of the material's genome. I want to go back to this premise that the fastest route to new materials is through miniaturization, because that's a really powerful observation. To understand the importance of that, let's take a step back and look at history. Entire ages have been defined by the discovery of new materials, whether we're talking about the Stone Age, the Bronze Age, the Iron Age, the modern superalloy age, or you could say we're living through the Silicon Age. New materials, new properties, new capabilities that advance civilization. That's basically the observation. Now, the problem is our route to finding new materials has been relatively slow. Stokia is based upon several key observations. The first and foremost is that new materials enable new applications across industries, regardless of what we're talking about. For example, the petrochemical, the energy, or pharmaceutical industries. They need new materials, primarily catalysts, to affect the processes that are important to them. The problem is finding these types of materials, catalysts in particular, can be very slow, taking decades sometimes, and very costly. So Stokia aims to use new innovations in nanotechnology to dramatically reduce both the time and cost associated with finding the materials that ultimately matter. And the premise is simple. Accelerated discovery will lead to greater competitiveness and profitability for their clients. And so it's a partnership type of driven model. We're creating a capability that allows partners to rapidly find the materials that matter. Example opportunities: CO2 to high-value chemicals, H2O to H2, methane to methanol, just to name a few. The problem, though, is the number of possibilities is almost endless. When you just look at the combinations of metallic elements, for example, in the periodic table, you combine that with this observation that it's not just composition that matters. On the nanoscale, it's both the size and the shape. At fixed composition, you have different properties, and even structure. So you take a spherical particle at a fixed composition, the way you partition those elements can have a dramatic effect on the properties of the resulting materials. So existing technologies are too slow and ineffective in accessing the material's genome. The material's genome not just defined by composition, but size and shape and structure. So Stakeholder takes advantage of technology we invented at Northwestern that allows you to create what we call mega libraries. Through miniaturization, we can make libraries of individual particles. Each feature is one particle type that is designed to systematically change in terms of composition and size, vary in terms of composition and size. Those libraries can be enormous. You can have upwards of billions of different particles spanning square centimeter areas. Scale is important here for two reasons. One is miniaturization means different properties with the same composition, but also the idea of a mega library becomes possible. If you had a mega library based upon macroscopic materials, it would be the size of a football field, effectively impractical. So once we build these libraries, we now can study the different features and look for materials that have interesting and useful chemical and physical properties. And as I said, catalysis is a big early goal in this particular case. We think this is going to allow us to accelerate how we find catalysts that are critical, certainly to the chemical, automotive, and petrochemical industries. A key partner in this is another company called TerraPrint that came out of the Institute. And they commercialized yet another innovation, and that was the idea of cantilever-free scanning probe lithography. The idea that you could create scanning probe tools with literally millions of tips that can locally control chemistry and deposit features that are on the nanoscale, allowing us to essentially build mega libraries routinely. These types of tools are commercialized. There's something called the TerraFab M and E series. They were commercialized by TerraPrint. They're now used routinely, primarily for research purposes around the world. They're on 3 different continents and in dozens of different laboratories. The reason they're important here is we can combine these with inventions out of the Institute that involve the generation of particles from what are called nanoreactors. Each of the tips can be used to deposit a polymer carrying a combination of metal salts in a fixed volume, a nanoreactor. Those reactors are very, very tiny in terms of volume. They can be down to yoctoliters in terms of volume. And when treated under the appropriate conditions, reducing conditions at elevated temperature, you can blow away the nanoreactor, reduce the metal ions, and all the metal atoms then coalesce to form tiny particles. A single mega library contains more new materials than have been characterized to date. That's really exciting. So in terms of ultimately addressing this challenge of interrogating the material's genome, this is one of the world's greatest tools. Here's an example of a particle library consisting of particles that have as many as 7 different elements within them. The vast majority of these have never been on the planet before. So this is an exciting way to, one, do research, but also to identify new materials that matter rapidly. To make these types of structures serially, incredible investment of time and money, and frankly not worth it until you've identified structures that ultimately make a difference. Now to end, I want to give one final example just to show you the reach and the impact of nanotech research. And it's called Azul 3D. This is a 3D printing company. Ironically, we were trying to develop through the technology that I'm going to talk about, the world's smallest 3D printer. In the process, we figured out how we can make the world's largest and highest throughput 3D printer. And through that innovation, we figured out that we could now begin to use this technology to transition 3D printing from prototyping to mass manufacturing. And effectively, not just taking on other 3D printing, but taking on injection molding in certain areas. And that's a really big claim to fame. And if we're successful, could dramatically impact how things are manufactured writ large. The technology I'm talking about is called HARP, or High Area Rapid Printing. And it is very high throughput, fast, near limitless in terms of scale. And you effectively produce structures that are one solid piece from almost a limitless palette of materials. And I show here the printer, the early printer actually working. Pretty exciting. You can see this lattice network being generated. Light is pulsating through a vat of resin, which has a window as a base. And you're seeing the object being printed on a metal plate that's being lifted out of that resin. So initially you print on the plate, and then you just continuously print off the structure that's being generated. And the fact that you can do it continuously makes it very fast. Now, the reason that this is so important is we've solved some of the biggest problems in the whole 3D printing area that enable this limitless type scale. So here's the premise of Azul. With this innovation, we can take on some of the biggest challenges in 3D printing. We've all been enthralled with 3D printing because it allows you to do things without a mold. Customization and complexity are not a problem, and you can have localized manufacturing. But the biggest limitations are it's too slow, too small, and ultimately produces parts that are weak and brittle. For that reason, it's been primarily used for prototyping purposes. Now, this is the landscape of 3D printing companies, at least as we see it. And they can be broken down into categories defined based upon the accessible printable area and speeds. Lots of companies that can print either small or large objects very slowly. And one company has emerged, Carbon 3D, that allows you to print relatively small structures relatively fast. And if you look at this, of course, the upper right-hand quadrant is empty. And that's where Azul really fits in. So with HARP, they have 100 times the speed of the slow players, and they can print 10 times, and that's going to go larger very soon, the area of a carbon. And that opens up the capabilities to think about taking on this issue of manufacturing, regardless of scale. Carbon was very interesting in the sense that it came up with a really slick innovation, which was to utilize something called the oxygen dead layer to effectively create a layer where polymerization doesn't occur on the other side of the window, so that you could print continuously onto a moving metal plate. The problem with this is you need a window that's semi-permeable with respect to oxygen, and you have to actively drive oxygen into that particular layer so that you can affect the process. As a result, cooling has to take place from the outside, and therefore you're limited in terms of area and frankly also speed, because the larger you go, the more heat that's ultimately generated. We came up with what we call the fluid flow interface, where we can actively move a fluorocarbon over the window and utilize that for 4 different purposes. 1, to create a non-stick layer. 2, to actively remove heat as it's generated and drive that liquid through a chiller. 3, filter it so that we are constantly generating a pristine interface. And 4, this works by establishing what we call a slip layer. So at critical speeds, you generate this non-stick interface, which is essential. And that also ultimately allows you to get the quality of parts that we ultimately generate and drag resin in so that you can compete with these rapid rates associated with this high throughput type process. And so that's a really slick invention because it ultimately allows you to completely transform the way we think about the 3D printing industry. And maybe this slide shows it best. So if you think about this, almost all 3D printing companies must make their money off of hardware. That's true of even carbon. But with HARP, you can begin to move the focus on resin. So if we look at how much money is ultimately made on hardware versus resin, you can see the purple is effectively resin. With these high throughput printers, if people are using them 24/7, the cost of the printer is almost negligible. As long as a certain amount of resin is used, that's where the money is ultimately made. So this is really critical, not just from a scientific standpoint. It completely changes the business model and how you think about effectively developing a company. So at the end of the day, we're not just interested in selling printers. We want to sell printers that are actively used for manufacturing purposes. And at the end of the day, we want to work with our partners to develop a model that really amplifies and bears this out. Examples are shown here where we can create parts in the medical industry, automotive industry, industrial and military, sports equipment industries. We have already a partnership that was established with DuPont, and very soon we will be announcing a partnership with a major player in the sports equipment arena as well. So I think we're really in a strong position now to capitalize on a lot of the early innovations in nanotechnology across many different sectors. To conclude, let me make a few acknowledgements. I'd like to recognize my research group at Northwestern. They're responsible for many of the discoveries and inventions that I talked about today. Support from really many agencies over the last couple of decades have made all of this work possible. And of course, the outstanding staff from the different startup companies that I mentioned throughout the presentation. They helped put much of this together. Thank you very much. I'd be happy to answer any questions.